Hello, my name is Megan Toler, and I'm an undergraduate senior at the University of North Carolina, Greensboro. And today I'll be telling you about how the plant Arabidopsis thaliana responds differently in a ground-based microgravity simulation study. Plants on Earth have adapted to light and gravity conditions in order to survive. Since light and gravity work simultaneously to affect plant development, spaceflight experiments are needed to separate these effects. In 2013, my lab did phototropic studies on the International Space Station, and novel plant responses to red and blue light were discovered. On Earth in 1G, the shoots of the plants, which are the top green leafy parts, have a positive response to blue light, meaning the shoots of the plants grow towards blue light. The roots, or the bottom parts of the plants, have a negative response to light, meaning the roots grow away to the light. Under red light conditions, roots and shoots have no response to the light condition, meaning it goes straight up and down with no curvature towards the light. However, in space, under 0G conditions, plants responded differently to each light condition. Under red light, plants had positive responses in both the roots and shoots, and under blue light, the roots now had positive responses. This is illustrated in Figure 4 on my poster. You can see on the left side that there is 0G, and on the right side there is 1G, and that is just a comparison of the responses that we see in space versus on Earth. These were amazing discoveries, and this brought about many new questions about how plant development happens under microgravity conditions. However, a big problem with this is that spaceflight is time and cost ineffective. To address this problem, my study goal is to perform a ground-based control experiment that simulates the effect of microgravity by using a 2D clinostat and compare these responses to spaceflight experiments. In other words, can I reproduce microgravity for phototropic studies on Earth? For the model organism, I used Arabidopsis thaliana and 3 different genotypes, wild-type LER and 2 phytochrome-lacking mutants, Phi-A and Phi-B. I surface sterilized and plated these seedlings and followed the growth timeline that was used on the spaceflight experiment my lab conducted. In the beginning, the plant is set under various dark and light conditions to promote germination, and then are set on the clinostat for 44 continuous hours. The clinostat is wheel-like and randomizes gravity for the plants by rotating at about 1.25 rotations per minute. You can see in Figure 3 an image of the 2D clinostat that we use in the lab. Once the data was collected, I took snips of the videos and measured the shoot and root angles with a software called ImageJ. I'm still working on analyzing the data I've collected, but we can make some preliminary observations based on the final images taken of the plants. The images shown in Figure 5, A and B, are the final images taken of our study. In Figure 5A, you can see that under red light conditions, the plants grow based on where they are on the plate. So the closer they are to the light, they have more curvature towards the light. And the farther away, they have a curvature away from the light. And in the middle, they have no response to the light. In Figure 5B, we can see in response to blue light on the clinostat that the plants show more of a response that we see on Earth. So you can see the shoots grow towards the blue light, so they have a positive response, and the roots grow away from the blue light, meaning they have a negative response. While the 2D clinostat seems to be a poor proxy for microgravity, this opens the door to many new avenues of research to be conducted for producing microgravity on Earth. One route that is being researched is using a 3D clinostat or random positioning machine to simulate the effects of microgravity on plants. Moving forward, these types of studies will give us insight on how we can grow in microgravity conditions like we see on the Moon and in Mars. Thank you so much. Take care and goodbye.